In the last video, you were introduced to the fetch command. In this video, we'll dig deeper into the RCSB protein databank and what the PDB ID contains. We'll also cover property selectors, which is a way to describe parts of the structure. But first, we'll go over some history of the RCSB protein databank. RCSB stands for Research Collaboratory for Structural Bioinformatics, and it was created in 1971 containing only 7 structures. Those structures included myoglobin, chymotrypsin, and lactate dehydrogenase. In 2003, RCSB was made publicly available, and over time, several thousand structures have been added to the database. At the time this video is being recorded, there's more than 180,000 macromolecular structures in the database. To find a specific structure in the database, you can search for the official name for the structure using the search bar. You have to always use the English name. We want to search for the protein carbonic anhydrase. By typing carbonic anhydrase, a drop-down menu will appear with suggested results containing the name. By clicking carbonic anhydrase in the drop-down menu, you avoid the result that just contains the name in its description, such as carbonic anhydrase inhibitors. If you use the advanced search, you could also search by amino acid sequence or molecular formula. As you can see, there are 65 results for carbonic anhydrase. Had we not chosen to click on the drop-down menu, there would have been more than 22,000 results. Under refinements, you can limit your search even more, for example by organism, experimental method, or resolution of the structure. We only wish to look at structures from eukaryotes, so we tick off the box and click the green play button. There are now 28 structures for carbonic anhydrase in eukaryotes that all look slightly different. Researchers may have determined the structures in different conditions or with differing methods. That, for example, could be with or without ligand, with or without mutations, and in different solutions. Sometimes, these differences cause differences in structure, even though they have the same amino acid sequence. For every structure, there's a corresponding PDB ID, title of, and authors of the scientific article that described the structure, release date for the article, experimental method, organism, name of the macromolecule and its classification, and any ligands. We'll now look further into the structure for carbonic anhydrase in a well-known model organism, Saccharomyces cerevisiae, commonly called baker's yeast. If we click on the PDB ID, we enter a page containing more information about the structure. This page contains general information about the structure, such as enzyme classification, which for carbonic anhydrase is the enzyme type called lyase. The expression system is the organism used for expressing and purifying the protein. In this case, it's E. coli, and we can see that it does not have any added mutations. Further down the page, you can see more information about the quaternary structure, stoichiometry, and links to the paper. There's also information about the protein structure and ligands. And lastly, there's information about the experimental data. Under 3D view, there's a more primitive version of PyMOL called JSMOL. JSMOL still have some useful functions, so in some situations you can avoid opening the structure in PyMOL. The annotations page gives information about other databases containing further information about the protein. This could, for example, be the website CAT, which contains information about tertiary structure. The experiment page gives further information about the experimental data. Under sequence, you can find information about secondary structure, and it gives an overview of the amino acids involved in ligand binding. The genome page shows the location of the gene in the given organism, here being the baker's yeast genome. To review the structure in PyMOL, you can either download a PDB file, or you can simply fetch the structure with a fetch command followed by the PDB ID. But what information does the PDB ID actually contain, and how is it read by the PyMOL program? Simply put, a structure file contains a list of points or atoms which has a x, a y, and a z coordinate. By calculating the distance between the points, the PyMOL program decides if the bonds are covalent or not, and in this way the structure is built in this session. The most important selectors, which all atoms have, are segment, chain, resin, resi, and name. When you click on an atom, the log will show you a name made up of 5 property selectors separated by a slash. Starting from the right, the first property selector is the atom's name. This is normally an abbreviation of the element, but often it also has a Greek letter. In PyMOL, there's no Greek letters, so you see that alpha is abbreviated to A, beta becomes B, gamma becomes G, delta becomes D, and so on. In this case, we've clicked on the residue carbon alpha, and it therefore has the name Ca. There's also a property selector called element, which is always only the abbreviation of the element. In the case of carbon alpha, the element property selector would be C. The next 2 property selectors in the line is resin and resi, separated by an apostrophe. Resi is the number residue in the chain that the atom's part of, and resin is the 3-letter or 1-letter abbreviation of that residue. Here we see a 3-letter abbreviation for lysine. The next property selector is the chain selector. Every continuous chain will have its own chain selector, which is usually a letter. The last property selector is the segment selector. This is usually only used in big complexes with many chains. In smaller structures, the segment and chain selector will often be the same. Lastly, the leftmost part of the name is the object name. By default, this is usually the PDB ID, but you can also have objects with other names. PyMOL has a number of additional selectors which allow you to select atoms by specific properties. This is, for example, formal charge or partial charge, which registers the charge of the atom. SS, which defines the secondary structure, and main chain and side chain, which defines the atom as being part of the main chain or the side chain. Note that the carbon alpha is defined as part of the main chain and not the side chain. Property selectors are extremely useful when you begin to write your own PyMOL scripts. This is because it makes it possible to select atoms based on, for example, what chain it's part of, what charge it has, what type of side chain it's part of, and many more.